Hi, I'm Kamil, the CTO and one of the founders of intoDNA, and also one of the co-inventors of sSTRIDE, the technology for direct and super sensitive detection of DNA breaks. In this presentation, I would like to introduce you to our DNA damage response functional assays. This is a group of assays that allow to visualize and then quantify DNA break associated with certain DNA repair proteins, and by that to understand better the biology of DNA repair or the mechanisms of action of DNA interfering compounds. We'll start with a short introduction into sSTRIDE, our technology platform on which all other assays are built on. sSTRIDE stands for Sensitive Recognition of Individual DNA Ends, and this is what the technology does. It enables sensitive detection and fluorescence labeling of free DNA ends occurring at the sites of DNA breaks or DNA fragments. sSTRIDE has some important characteristics. It is direct, which means that the detection occurs in intact cell nuclei in situ at the site of a DNA end with no dependency on DNA repair processes. It is quantifiable. The fluorescence foci formed at the sites of DNA breaks are easily distinguishable, and our in-house built algorithms enable their objective quantification. sSTRIDE is compatible with different biological materials, including FFP tissue sections. And finally, sSTRIDE is sensitive and specific, capable of detecting individual single or double-strand DNA breaks of different types, depending on which assay variant is chosen. To understand how our DNA damage response functional assays work, it is essential first to get familiar with our basic STRIDE assays, which detect the total pool of DNA strand breaks. As you can see in the schematic diagram, STRIDE is a method, which means there is a sequence of steps that need to be followed to obtain a signal as the outcome. The first step in each of our assays is enzymatic incorporation of nucleotide analogues to DNA ends. In the case of dSTRIDE, which detects DSBs, this first reaction is performed with the use of a terminal transferase. In step 2, a pair of primary antibodies is attached to the labeled DNA ends, which is then followed in step 3 by binding secondary antibodies and signal amplification using an RCA reaction. In the case of sSTRIDE, which is a method for detection of SSBs, the steps are very similar, with the main difference in the initial enzymatic reaction, which is now carried out by polymerase I. sSTRIDE and dSTRIDE assays allow to detect DNA strand breaks, no matter whether they are under repair or where they are localized within the nucleus. Our DNA damage functional assays, on the other hand, can be used to narrow down the detection only to those breaks that are associated with a certain DNA repair protein. So how this is achieved? As you can see in this diagram, again, 3 major steps are needed to obtain a fluorescence signal. This time, however, one of the antibodies used in step 2 is directed against the protein of interest. The second antibody still binds to the modified nucleotide analogues introduced in the enzymatic reaction in step 1 at the site of a double or single-strand DNA break. As a result, the signal will only appear in those cases where the protein of interest and DNA strand break are in close vicinity. sSTRIDE-based DNA damage response functional assays are built on the same principle. Here is a list of DNA damage functional assays that we introduced so far. Each of the assays' name contains the name of the protein that is being detected. For example, there is the sSTRIDE PMS2 assay that detects PMS2, a protein known to be engaged in mismatch repair at the sites of single-strand DNA breaks. On the left, you can see the list of assays built on our sSTRIDE, like sSTRIDE PMS2, SMUG1, and RPA70, and on the right, similarly, assays built on dSTRIDE basic variants, such as dSTRIDE RAD51, RPA, or WRN. Importantly, with our expertise, it is possible to customize the existing assays to suit your needs and also develop completely new assays, targeting the protein of your interest. One of the first DNA damage response functional assays that we developed, fully validated and introduced was sSTRIDE PMS2. As you can probably guess by now, this assay allows to detect PMS2, a protein that is one of the crucial players in mismatch repair, in the vicinity of a single-strand DNA break. On this slide, you can see representative images of U2S cells in which MMR-related SSBs were visualized using this assay. This is compared to an image of a cell of the same type where SSBs were detected using our classic sSTRIDE assay. On the right-hand side of this, and also in the next slides, we present a part of the validation dataset. First, we looked at whether the signal in the sSTRIDE PMS2 assay can be modulated by treating the cells with 6TG, an agent known to induce MMR. As you can see, we were able to see a significant increase in the readouts after the treatment. Importantly, an isotype control sample yielded very low readouts, confirming the specificity of the assay. This is compared to readouts for the classic sSTRIDE assay, which naturally are on a high level. An isotype control is not the only one that can be applied in such an assay. We decided to expand the scope of validation and also look at scenarios where either both antibodies, which are necessary to conduct the assay, or only one of them is omitted. The results can be seen in this graph. The readouts for each of the controls are expressed as a percentage of the readout in the full procedure. As for positive controls, we further tested 3 different concentrations of 6TG and could see that the sSTRIDE PMS2 assay performs in a dose-dependent manner. This can be seen on the right-hand side of the slide. In the final part of the validation, we wanted to include a biological negative control. To this end, we used HAP1 isogenic cell lines, a wild type, and a PMS2 knockout. As you can see on the histograms here, which present the percentage of cells with a certain number of sSTRIDE PMS2 signals, these two cell lines are strikingly different. In the PMS2 knockout cell line, more than 90% of nuclei display no signals at all. Finally, we tested the performance of our new assays when the MMR pathway was modulated using 3 different inhibitors targeting PMS2, again in HAP1 cells. What we could see was very encouraging. All 3 MMR inhibitors reduced the number of sSTRIDE PMS2 positive nuclei when compared to untreated cells. Another interesting assay that we recently introduced, sSTRIDE SMUG1, can provide insights into the BER pathway. SMUG1 is a DNA glycosylase which removes damaged or inappropriate bases from DNA, creating an abasic site. We based this assay on our classic sSTRIDE variant. To test if our assay is able to detect the recruitment of SMUG1 during the DNA repair processes, we treated HAP1 cells with 5-hydroxymethyl-deoxyuridine. We were able to show that after 24 hours from the treatment initiation, the level of sSTRIDE SMUG1 signals increased. Again, the isotype control, which is also shown on this graph, was at a low level. In the next experiment, we verified if SMUG1 knockdown influences the assay readouts, and the answer was yes. When SMUG1 siRNA was used, both the baseline and the responses to HMDU treatment were on a significantly lower level than the control cells with intact SMUG1. Last assay that I want to mention today, and at the same time probably the most interesting one, is called dSTRIDE RAD51. It enables detection of RAD51 at the sites of double-strand DNA breaks, so it can inform about the activity of homologous recombination DNA repair pathway. Similarly to what was shown before for sSTRIDE PMS2, here we also see some assay validation data. dSTRIDE RAD51 signal levels were elevated in uterus cells after treatment with bleomycin and etoposide, and negative control were kept at a satisfactory low level. Because RAD51 is such an important protein in the HR pathway, its absence usually means that the repair pathway is defective and the cell is defined as HR deficient. The status of HR is very important for certain cancer treatment strategies. HR deficient tumors can be treated with PARP inhibitors, leading to the targeted killing of cancer cells. The classic way of defining the HR status of cells is to look for mutations in BRCA1 or BRCA2 genes, or perform IF or IHC detection of RAD51. These cellular assays, however, are limited in their sensitivity and specificity. The sole accumulation of RAD51 in the nucleus of cells doesn't necessarily mean that the pathway is active. dSTRIDE RAD51, on the other hand, is quite different. It allows to quantify RAD51 only when it sits on the DNA break. It is a functional assay. We wanted to test whether the dSTRIDE RAD51 can be used to distinguish between patients with different status of BRCA genes. In a joint effort with MSKCC, we performed dSTRIDE RAD51 labeling of biopsies taken from 6 cancer patients. And the results we obtained were very striking. We could see a clear difference in dSTRIDE RAD51 readouts level between patients with mutations in BRCA1 or BRCA2 genes when compared to patients that had no mutations. This result, although on a limited dataset, provides proof that dSTRIDE RAD51 has the potential to become an important improvement in the assessment of HR status of cancer cells. Thank you for your attention. I hope you found it interesting to listen about our unique DNA damage response functional assays, and that maybe one day you would be willing to use them in your research or drug development related activities. Thanks for watching.